They're mitoses.
No, they're mitoses.
I had to read the book pretty closely, and it hurt my eyes. So then I put it a bit further away.
Yeah.
And it said, mitoses are definitely mine.
Well, I did a bit of a diagram, Wayne, to see how much of mitoses actually were. Turns out, mine, mitoses.
Well, there you go. Explicit proof. You win this round.
Conclusive.
Well done. Well done.
I mean, they're not quite as... That's not my foot. That looked quite scary.
Yeah, pretty demented.
Yeah.
So, the cell cycle is the point of this.
Yes, and that's when you get in your bike and you hurt yourself, but then you heal back.
Yes, so repair.
Yeah. That's what it's for.
And growth.
Repair.
So the cell cycle in general, treat it as a cycle.
Yeah, goes round, starts at one stage, gets back to that stage at the end.
And when it gets back to that stage, there'll be 2 identical cells, and then that keeps going, and then there'll be 4, and then 8, and then 16.
And eventually there'll be you. Hello.
Hi.
You're right.
You're right. Cool.
Yeah.
So this is the cell cycle.
Yeah.
Most of it we don't really talk about.
Yeah, most of it is quite dark and evil. Not really, but it's called the interphase. That takes most of the time, and that's when everything gets doubled.
Yeah. So everything's doubling in this interphase stage. So if we had a cell, we're talking about eukaryotic cells, and they have a nucleus, as we know.
We do.
In those nucleuses is chromosomes, as they're called at this point. And so they look like, we'll sort of draw them like this, little squiggles.
Little squiggles, alright. And what happens is everything doubles, including those, we end up with X's.
So by the end of interphase, I'm going to draw it as 2 separate squiggles, and we'll draw them as X's in a minute.
Okay.
So that should be that many in this scenario. So that everything doubles.
Yeah, which is pretty handy.
Now we focus majorly on this next point.
Yeah, this next bit is called mitosis, and that's mitosis.
His toesies,
Yeah.
that's right. And we divide this short section into 4 sections. So we divide just this bit up, so it's not the whole thing.
Yeah. To do much more detail, so the first part of this is prophase.
Yeah. So that's the first part.
So you're really, really blooming good at phasing right now.
Yeah. We then go on to metaphase.
Yeah.
I'm just going to put meta and ignore the phase from now on. They all end in phase.
So you met her, she was lovely.
She is Anna.
Yeah. Anaphase. And
you got a number, didn't you?
Yeah, I did. And I phoned her, and it was the wrong number.
Yeah,
That was
well.
sad. But I did use a telephone, so it's telephase next.
Telophase.
Yeah.
Prophase, metaphase, anaphase, telophase. Those are the
So what did you tell her?
phases. Oh, I told her to watch this video.
Oh, that's
Hi
good.
Anna.
How you
How you doing? Right.
Yeah, good.
Good.
Alright, so what happened in these phases, Wayne's question?
Quite a lot of things happen.
Alright.
Throughout interphase, the organelles and everything in this cell replicate. And you don't need to worry about that. We're focusing majorly on this nucleus here. So we've said that the chromosomes have all replicated. There's 2 of every chromosome.
There is.
Now in this prophase phase, we no longer call them chromosomes.
No, we call them chromatids.
I'll draw you a cell and a nucleus.
Oh, that's
and
very
you can draw
kind of
yourself
you.
some X's if you'd like.
Alright, and this is when they become the X's that I mentioned a bit earlier.
So we should probably colour code them.
I
So
will.
there was 4 before, there's going to be 4 now, but now there's going to be 4 chromatids, so they're bonded together as X's. Just like that. And where they bond together, this central point here, where all of these X's come together,
Yeah,
is a centromere.
that's pretty good. So what happens in our first phase, Wayne? Apart from that.
Well, apart from that, these chromosomes, now called chromatids, they get shorter than they were up here.
Yeah, bit that now.
They become X's joined at a centromere, and that's pretty much it.
Then the nuclear envelope breaks down as we enter metaphase.
That was silly.
Yeah.
Yeah, so sort of the end of prophase, beginning of metaphase, this nuclear part breaks down. So
Yes.
if we draw another cell, it's not going to have this anymore.
Yeah,
It's
we
just
just
an
have our
egg.
X's. But how are we going to have them?
Well, that's the part that's developing now. Inside eukaryotic cells, we mentioned in the first video when we drew it up, centrioles.
Now they're very handy in mitosis to make a mitotic spindle.
They do. The centrioles themselves go to sort of opposite poles and make like a north pole and a south pole sort of thing.
Yeah.
And then they make, we express it with dotted lines, as Triv said, a spindle like this. So if we've got 4 as an example, there'll be 4 here. And what these X's do, remembering this part is broken down, is they line up neatly in a neat long line, like you did in primary school for a lesson. You draw it up in a nice line along these spindle, just like that.
Just like that.
And that's lovely, isn't it?
That's really nice, Wayne.
And that's metaphase.
Okay. Then we have anaphase. Now this is when stuff actually starts splitting up.
It is.
So these 2 things then pull at our chromatids there, split them a little bit. Let's take a bit of drawing.
It is. Redrawing up the spindle as it was before.
So they're moving these bits are moving up a bit, these bits are moving down a bit. Essentially what we're going to do is try and get to how we had the started interface. So one of each of these in each cell.
Yeah, so there's now half an X going up and
Mhm.
half an X going down. I'm colour coding it so it's sort of back to this, but
Yeah.
separating.
Separating.
Talkway. Right.
Alright, so then we phone Anna.
We do. Hi Anna, how you doing?
Yeah, good.
Awkward. I'm going to have to split you in half.
Sorry about that.
So I realised that had two meanings after I said it.
Yeah.
Biology.
So what happens then is essentially cell division, but these are pulled to each pole.
So what, I'm going to go straight to drawing them as two. I'm going to
Yeah.
express it here. So it splits down the middle,
Yeah.
so that there's equal amounts on either side.
They split right into the poles, and then they reform the nucleus, and it makes two very happy cells like this. They look a bit like eyes.
So there's 4 blue ones in that one, there's
Yeah.
only 4 red ones in this one, because they haven't doubled up yet.
Mhm.
So that's 2 cells, they're called the daughter cells.
And yeah, there's only 2 from mitosis, 4 from meiosis, if you remember that from
Yeah.
GCSE.
We'll get onto that later in the course.
We do.
So these cells then instantly both undergo interphase, so they start doubling up immediately.
And that takes ages.
It does, that takes a very long time.
Mhm.
And there's a neat little experiment you could do. Probably you've done it in your lab, but it will come up in your exam probably.
Yeah. That's about root tips.
It is. So you take a root tip, you cut it off, very rude.
Yeah.
You then cut it up and be really small and stuff.
And you put some things which breaks down stuff.
Yeah, so hydrochloric acid is a typical
Yeah.
example, something like that will break up the walls a little bit, but not enough to completely destroy it. You then put in some sort of stain, there's lots of different stains you could use, like a
Yeah.
cetyl blue is the one I remember.
That's the one you would just see in your microscope.
Yep. There's also red ones and all sorts, and pink ones and all sorts. Yeah.
In random scarlet, black, and every
All of Andrew Lloyd Webber's colours.
All of them.
And then you crush it up lots, you put it in a slide, you put it under the microscope, and you'll be able to actually see the cells, and you'll be able to look at these spindles
Yeah.
and stuff and
You have
see.
to work out what cells are in each stage, and you'll notice that nearly all of them are interphase.
Yeah, so they're just doubling up and not a lot really going on.
Very exciting though.
Sort of, I think it's pretty cool.
Yeah.
So, these may be your toes, but this is my cup of tea.
Oh.